Hello and welcome to another episode of Old World Agriculture. In this episode, the Landraces of P. You'll start off with an experiment run by the University of Athens under the researcher Georgia Natatsi. This researcher is researching pea landraces because peas are one of the best nitrogen fixers in terms of agricultural legumes. They also have the highest protein content. In this study, researchers studying different landraces for their resilience against weed competition and insect damage, in hopes of utilizing different landraces to breed new varieties of pea for use in organic systems. The peas are planted in organic plots covered with different weeds, specifically including wild mustard, corn poppy, common henbit, ivy leaf speedwell, cleavers, common firmatoy, corn chamomile, common knot grass, and wild oats. 3 landraces were tested likewise within the system. They were tested against a common cultivar of pea called Onward. And here are the results. First, in terms of nitrogen content and nitrogen fixation, the amount of nitrogen fixation that happened in peas in all situations ranged between 45.2 kilograms per hectare and 125 kilograms per hectare. It was found that in organic plots that the cultivar Onward, the common commercial cultivar, fixed nitrogen much better in a conventional system than it did in an organic system, while the landraces did poorly. This was inverted within the organic system. From a protein content and yield standpoint, the farming system had no significant impact on yield or protein content. The common commercial cultivar Onward and the landrace strain AUAAND-RO001 had roughly identical yields to the commercial cultivar Onward in both systems. The above-mentioned landrace AUAAND-RO001 was found to be more tolerant of weed competition than the Onward commercial pea variety. This can be attributed to lower tolerance to colder weather found within the Onward genotype, indicating that this specific landrace is actually very cold tolerant and by proxy able to grow faster than the weeds in an Athenian growing system. This landrace, on the other hand, does have unique capability of high levels of genetic variation, causing variable levels of maturity for each individual plant. On a side note, the cold weather within the field may have been a key factor in why both that specific landrace and the commercial cultivar performed comparatively. Seed protein content within the landrace mentioned was far lower than what was found in Onward. The other 2 landraces tested had higher numbers of seeds, but the seeds were far smaller. It was concluded that the landrace AUAANDR001 is likely the best suited for breeding programs within Athens to improve pea breeding programs due to the resilience of the species in dealing with plant-to-plant competition and abiotic stresses. The researchers were unable to gather any data regarding insect damage, however. Another paper based in Cordoba, Spain, was made by the researcher Dr. Diego Rubiales. Within that country, there's a plant parasite called broom rape. It is found all across the Middle East and the Mediterranean basin, acting as a major constraint within pea production, alongside other legumes. The plant can parasitize other plants by attaching their roots to the roots of the legume plant to absorb nutrients. Breeding for resistance against broom rape is a complex genetic due to it being a quantitative trait, and has been only found in other species such as fava bean. And that resistance trait is gradually accumulated through different breeding programs combining different resistant genotypes together. And a similar effort has only recently started, as of the writing of this paper, for pea breeding process. Several landraces were tested for resistance to broom rape. These landraces were PS624, PS423, P665, P675, and P660. It was found that the hybrids between common cultivars and these landraces had incomplete resistance comparable to common resistant lines of P, but still well above susceptible lines in terms of resistance, with these P cultivars being able to induce root damage in the parasitic species broom rape. Other experiments along a similar line have been studied, which showcase that certain P landraces actually have the capability of inhibiting seed germination of this parasitic species, as well as inhibiting the parasitic root formation within that species. Several other studies have been done using these same landraces, several of which were derived from back crosses back into commercial cultivars having less lineage within the landraces, but still having similar levels of resistance to the broom rape parasitic plant, indicating that resistance may not be as complex as earlier suggested. This is for the landrace P660. Other breeding lines are being developed for P cultivars derived from combining the landrace PS624 with susceptible cultivars to improve their resistance. In most instances, the actual inheritance of these traits remain unknown. And as of the writing of this paper, no genetic studies have been done to showcase the nature of these genetics. But if it were to be shown, then resistance could be improved vastly to the level of faba beans. Now on to the next section. This is a paper by Silly Sun of the Chinese Academy of Agricultural Sciences based in Beijing, China. This researcher wanted to find resistance to powdery mildew, a common disease within China and many other parts of the world that affect pea. Until relatively recently, it was thought that powdery mildew in pea is only caused by one species, E. passae, but in later years it was found that other species such as E. trifoli and E. baumerli were also found to be disease-causing organisms that cause the powdery mildew disease. As of right now, only one species is known to have resistant hosts. Within the pea genome, there are pea varieties that have 3 genes, ER1, ER2, and ER3. The former 2 are recessive genes, and the last is a dominant gene. These 3 genes are additive, each one adding an additional form of resistance to powdery mildew, at least for that species. The gene ER1 is widespread throughout the pea germplasm and landraces and is considered stable against powdery mildew. The resistance gene ER2 is only harbored within a few landraces, specifically SVP951, SVP952, and JAI2480, as far as we know, as of the writing of this paper. The ER2 gene confers resistance to powdery mildew in certain locations but fail in other locations. The ER3 gene is a gene found in the wild relative of the pea, Pisum fulvum, and has successively been incorporated into the regular domesticated pea. All 3 genes have a different effect on the disease, making them complementary to each other. 2 recently discovered species of powdery mildew are already able to overcome the ER1 gene, and recent discoveries found that E. trifoli can overcome ER3, but not as of yet ER2. The goal of the paper was to figure out how to find new traits within the pea germplasm that confer resistance to these new species, and here are the results. The researchers screened 322 landraces to find out which ones were the most resistant. It was found that the pea landraces from Yunnan were amongst the most resistant to all the species of powdery mildew. The landraces from Yunnan were 33 in number. Within the 33 landraces, it was found that 12 of the landraces were immune to powdery mildew completely, 4 were highly resistant, and 17 were simply resistant. 11 of the 12 immune landraces were from Yunnan, as were 3 out of the 4 highly resistant landraces, and 4 of the 17 resistant landraces. This makes sense considering that Yunnan is a unique location with a wide array of different climates within that small region. This would have led to different mutations and by proxy different genes for resistance. Various subforms of the ER1 gene have been found here. So far, 6 variants of this gene have been found here that could be used to breed new varieties of P, most of which are developed through point mutations, causing a substitution or pair deletion mutation. Although one strain was created through the insertion of a piece of DNA from another source of the genome being added within that gene. So for breeding new powdery mildew resistant strains, it's likely that the Yunnan landraces would be a good spot to start. And that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.